Welcome to Biology My Passion. I am Soumya Harikrishna. We have been learning the chapter Biomolecules. We discussed all the biomicromolecules. Today we will discuss biomacromolecules. Before that, let's learn about metabolites. What are metabolites? Metabolites are the intermediary products produced during metabolic reactions. You know, there are lots of reactions happening in our body. They are collectively called metabolic reactions. So these reactions result in products. Those are called metabolites. These metabolites are again made into different substances required for our body. So here we can divide metabolites into two: primary metabolites and secondary metabolites. Usually in case of animals and in case of plants as well, direct metabolic reactions will result in certain compounds, organic compounds like proteins, nucleic acids, carbohydrates, etc. These products, all of them have got some very specific functions in the physiology of the body or the maintenance of the body. That means our life processes. For example, they are directly involved in reproduction, growth, development, etc. So they are called primary metabolites, which are the products of the metabolic reactions, and they have definite roles in the body. Whereas there are second group of metabolites present only in plants, certain microbes and fungi. They are also produced as a result of these chemical reactions, but we do not know what is their actual function in the physiology of that particular organism. But many of them are useful to us. Alkaloids, terpenes, nitrogen-containing compounds, etc. are examples. So they are not directly involved in growth, development, reproduction, etc. So these are the two types, primary and secondary. Some secondary metabolites are given in the table. First is pigments. You know that carotenoids, anthocyanin, these are pigments present in the plants. Carotenoids are involved in photosynthesis. Anthocyanin gives color to the plant parts. Now alkaloids like morphine, codeine, etc. Morphine is used as an analgesic in post-operative condition. Then terpenoids are monoterpenes, diterpenes, etc. Essential oils like lemongrass oil, these are present in many plants which have got very nice fragrance and they can keep away insects and all. Toxins like abrine, resin, etc. Some plants have toxins as their defense. Then lectins like concanavalin A, drugs like vinblastine, curcumin, etc. These are medicinal. Vinblastine is present in a plant called Vinca rosea and curcumin is present in a turmeric which has got an antiseptic property. Then polymeric substances like rubber, gum, cellulose, these are all economically important for us. Beginning of this chapter, we discussed the analysis method of biomolecules. We got two fractions. One was the biomicromolecule that is present in the acid soluble pool, whereas biomacromolecules were in the acid insoluble pool. So why they are in the acid insoluble pool? Because their molecular weight is more than 10,000 daltons. So which are the main molecules coming in that? These are polypeptides, polysaccharides and polynucleotides. What is meant by poly? Poly means many. So that means these molecules are made of many individual units, or we can say monomers. So polypeptide means this is an example of proteins. Polypeptide means it's a protein. So here suppose I draw this as an amino acid. So many amino acids are connected by peptide bond. So this bond between amino acid is peptide bond. So proteins are otherwise called polypeptides. Now coming to polysaccharides. Polysaccharides are sugars. In sugar, simple sugars like your glucose, fructose, these are all called monosaccharides. So when suppose this is a monosaccharide, then one more monosaccharide added, then we call it as disaccharide. But like this many are added, then it is called a polysaccharide. Polysaccharides are basically the carbohydrates we say. Now third is polynucleotide. So another molecule, polynucleotide means many nucleus. Suppose I am drawing just like this to make you understand. Many nucleotides are connected end to end. So we get the nucleic acid in this, that is DNA, RNA are examples of polynucleotides. So all the macromolecules are connection of many individual molecules with certain bonds. So now let's discuss one by one. Let's start with the polypeptide. What is another name of polypeptide? Proteins. As I mentioned, proteins are polypeptides. That means individual monomers or units are connected, n number of units are connected by peptide bonds. What are those units? They are nothing but amino acids. So amino acids, usually 20 types are there. They fall into two categories: essential amino acids and non-essential amino acids. Some amino acids are very essential for our health, so we have to obtain them through the dietary protein. That means through the food that we are eating, we obtain these essential amino acids which are very crucial for our health. So they are called essential. But there are certain amino acids which our body can synthesize as per our need, that is called non-essential amino acids. Now, you must have heard of homopolymer and heteropolymer. So all the individual units are the same, then it is called a homopolymer. Look at this beads. These beads are all alike. So many beads are there, we can call it as a polymer. Each one is a monomer, and if all of them look alike, then we can call it as a homopolymer. Whereas in the second chain I am showing you, it has got different types of beads in it. First maroon beads are there, then gold beads are there, then blue beads are there. So since there are many different kinds of monomers, they are called a heteropolymer. So when we talk about proteins, they are not monomers or the same monomers, homopolymer, they are heteropolymers. That means different types of amino acids are there. We told 20 different types of amino acids are there. They are arranged in different order to make thousands of proteins required for our body. That's why I use different colors here. Now coming to the functions of proteins, they are doing a lot of functions in our body. They act as the agents for facilitating the transport of nutrients across the cell membrane. So many substances move in and out of the cell membrane with the help of proteins. Second function of proteins is they fight infectious organisms. That means whenever we have infection in our body produces antibodies. These antibodies are actually kind of proteins called immunoglobulins. Then we have some hormones like insulin and all, they are protein hormones, or majority of enzymes in our body are also made up of proteins. This table in your textbook shows some proteins and their functions. Collagen is actually the most abundant protein in the animal world. Intercellular ground substance means it binds the different substances, different cells together. Then trypsin is the enzyme. Last year you learned in class 10, it digests protein. Then insulin is another hormone which is protein hormone, and also as we mentioned fights against infectious agents, antibodies or immunoglobulins. Then sensory receptors are like a smell, taste, then hormones, etc. For example, we learned about olfactory receptors, gustatory receptors, etc. last year. And finally, GLUT4. GLUT4 stands for proteins which enable glucose transport into cells. GLUT4, glucose T transport. So you can easily remember GLUT4 for glucose transport. Now let's talk about the structure of proteins. When chemists discuss the structure, they take it in a two-dimensional way. Physicists will go to three-dimensional way, but biologists take it in a four-dimensional view. That means proteins can have four-dimensional structures. First is called a primary structure. In primary structure, the amino acids are just connected end to end to get a linear chain, starting from the first amino acid on the left side and the last amino acid on the right side. The first amino acid has got amino group to the left side, so we call it as N-terminal, and we know that the right one must be ending with a COH group at that end, that is called a C-terminal. Now if I show you with the help of a wire, you know that this wire, it is considered as a primary structure. Now if it is coiling in a right-handed fashion in the form of a spiral staircase, then we call it as secondary structure. Now if it is coiling in such a way that it's becoming a hollow ball, then we call this as the tertiary structure, which has got many crevices or pockets to which the other substances can bind. We will learn about it. This structure is called the tertiary structure, and this is very important for biological activities. Now like this, if more than one comes, then it is called a quaternary structure. As you can see in this picture, this is the molecule of hemoglobin, which is an oxygen carrying pigment in our body. It has got 4 tertiary units connected together. They are called the alpha chains and beta chains. 2 alpha chains and 2 beta chains are present. The next biomacromolecule is polysaccharide or carbohydrates. They are otherwise called glycans. They are 2 types, homopolysaccharides or heteropolysaccharides. As I told, homopolysaccharide means same kind of monomers will come. Heteropolysaccharide means different types will come. So both can be present in the carbohydrate category, or they can be branched or unbranched. So if you see cellulose, starch, glycogen, inulin, these are all homopolysaccharide, means their individual units are same. For example, cellulose. Cellulose is a homopolysaccharide made up of only glucose molecules. You know that cellulose is actually a structural component of the plant, means it makes the cell wall. For example, the cotton thread that you are getting or the paper pulp we are making, all these are cellulose because it is made up of plant cells. So it is a structural component. Whereas when you come to starch, starch is a storage carbohydrate in plants because excess glucose is stored in the form of starch. So it is also made up of glucose. Another variant of that starch is present in animals. We cannot store it in the form of starch, but we store it in the form of glycogen. That is another homopolymer of glucose. That is also a storage carbohydrate. Another storage carbohydrate is inulin. Inulin is made up of mini fructose. It is only present in plants. We are not able to digest it. Then when you take a carbohydrate, it can have 2 ends. The right hand end is called the reducing end because there is an aldehyde present in it. The left hand end is the non-reducing end. No aldehyde is present. And yet another complex polysaccharide means it consists of many derivatives of these monomers. Then we call it as chitin, which is actually making up the cell wall of fungus, or we know the exoskeleton of insects are all made up of chitin. And chitin is also a homopolymer. But the difference here is cellulose and chitin are structural polysaccharides, whereas starch, glycogen, inulin are all storage carbohydrates. In this picture, you are seeing the cartoon structure of a branched chain of the starch. It has got many loops in its structure which can hold iodine. You know that whenever we want to prove the presence of starch, we add iodine to it. It turns blue-black, means starch is present. Instead of starch, if you take any other kind of saccharides like sucrose or fructose, glucose and all, if you add iodine also, it won't show color change. Why? Because they don't have the loop to hold the iodine molecule. The next macromolecule is a polynucleotide, or we can call them as nucleic acids. We learned already the nucleic acids can be of 2 types, DNA or RNA. So since they are polynucleotides, their individual units are nucleotides. Each nucleotide has got 3 parts. 1 sugar is there, 1 nitrogen base is there, and 1 phosphoric acid. All 3 together we call as nucleotide. So usually it is seen like this. There will be a sugar, then a phosphorus. From sugar, a nitrogen base will be projecting inwards. If it is RNA, there is only 1 strand like that. Again the same unit repeats, SNP. Again SNP. So only 1 strand will be there for RNA usually, but DNA means sugar, phosphorus, sugar, phosphorus, sugar. From every sugar again 1 nitrogen base will be popping inside. So these nitrogen bases are connected by hydrogen bonds. So 2 strands are there. Always backbone is made up of sugar phosphate and there is a nitrogen base. And this unit is nucleotide. So repeating nucleotides are there in case of the nucleic acids or polynucleotides. So here sugar and phosphorus are connected by ester linkage, whereas sugar and nitrogen bases are connected by N-glycosidic linkage, and 2 nitrogen bases are connected by hydrogen bonds. We learned about nitrogen bases already in detail. Sugar also, they are actually monosaccharides. They are of 2 types, ribosugar or deoxyribosugar. If it is RNA, ribosugar is present. If it is DNA, deoxyribosugar is present. Then when we come to nitrogen bases, as we already discussed, they are purines or pyrimidids. They are heterocyclic compound. Then phosphoric acid is the phosphate group, which is H3PO4. Hope you understood all these concepts very well. If so, please like, share and subscribe to my channel Biology My Passion. Thank you for watching.